Hello and welcome to my presentation on the cell and cell division. This is for Biology 2401, AMP1. The main parts of the cell are going to be your nucleus, plasma membrane, and cytoplasm. The nucleus holds the DNA makeup of the cells, and when the cell is not dividing, the genetic material are in the form of chromatin inside the nucleus. The plasma membrane separates the inside of the cell from the surrounding structures and creates a protective barrier. Here you can see the plasma membrane of each cell shown. The cytoplasm houses all the contents between the other two, and floating in the cytoplasm are the cell's organs, also known as organelles. Organelles of the cell are going to be the ribosomes, the rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, mitochondria, and centrioles. Also are cytoskeletal elements, which are microfilaments, intermediate filaments, and microtubules. Ribosomes are tiny sphere-shaped organs that are composed of RNA, floating or attached to a structure within a membrane, typically the rough endoplasmic reticulum, and this is usually the site of protein synthesis within the cell. Rough endoplasmic reticulum, pictured here, is studded with ribosomes, and the tubules provide a place for storage of the proteins made by the ribosomes to other parts of the cell. Smooth endoplasmic reticulum, which you can see in this picture, looks almost like the rough ER, however, it just doesn't have the ribosomes on the outside. Is a site of steroid and lipid synthesis, metabolism of lipids, and drug detoxification. The Golgi apparatus are flattened sacs with bulbed ends, and they're usually found close to the nucleus. They assist in packaging proteins and other substances to get them ready to come into or leave through the plasma membrane. These are going to be the bulbed ends that it talks about. Lysosomes carry digestive enzymes, and they digest worn out organelles, and they're also known as suicide sacs. These little boogers right here have the ability to completely destroy an entire cell, which is why they're known as the suicide sacs. Peroxisomes look a lot like lysosomes, and they just detoxify alcohol and any other harmful chemicals that make their way into the cell. These are mostly found in the kidneys and liver, so they can detoxify the alcohol. Mitochondria, pictured here, is rod-shaped. It has double membranes, as you can see, an outside membrane, and this is an inside membrane. The inside membrane is in folds, and it oxidizes food to make ATP, which is the energy for cell uses. It's also known as the powerhouse of the cell due to it making all the energy. Centrioles, which are here, are always paired in twos, so you'll have this and another set, and each two sit at right angles of each other. They usually have 9 triplets of microtubules, which you'll see later, and they direct the formation of the mitotic spindle during cell division, which we'll discuss a little bit later in the presentation. The cytoskeletal elements include microfilaments, which consist of actin, which is right here, intermediate filaments, which are the microtubules, intermediate filaments here, and then microtubules, which are pictured here. And this is just a picture of the entire cell body with all the organelles on the inside. You can see the rough ER here and here. The Golgi apparatus is here. The smooth endoplasmic reticulum's here. Lysosome here. Mitochondria here. Nucleus, nucleolus. The purple inside the nucleus is the cytoplasm. Next, we're going to talk about cell division. In cell division or mitosis, you have interphase, prophase, metaphase, anaphase, and telophase. Interphase, which is the first part, is actually not part of the mitotic process, but it's where most of the cell spends most of its time. Interphase has 3 different parts to it. G1, which is growth. S, which is growth and DNA synthesis. And G2, which is growth and the final preparation for division. The next phase is prophase, which is where chromatin condenses and forms into chromosomes. Each chromosome has 2 identical sister chromatids that are held together in the middle at the centromere. As the chromosome appears, the nucleoli will disappear, and a pair of centrosomes will move away from each other, which here you see the chromatin condensing into the chromosomes. You have the 2 identical sister chromatids held together with a little dot right there, which is going to be your centromere. Over here you have the centrosomes, which are separating and starting to form microtubules, which will help in the separation of the cell. In metaphase, the centrosomes are on opposite sides of the cell, and the chromosomes form a line down this middle of the cell. Enzymes begin to separate the chromatids from each other at the centromere. And then you move over into anaphase. Anaphase is the shortest phase of mitosis. Microtubules pull the chromosome towards the pole it faces, and they'll form into little V-shaped chromosomes as they pull. Also, as they're pulling apart, these are pushing each side of the cell apart to help with the division of the cell. If the chromosomes were to stay in the chromatin form, instead of having these real dense V-shaped chromosomes, you would have tails that would tangle and tear as they pulled past each other, which is why they condense into the shorter chromosomes. Telophase, which is the final phase of mitosis, starts when the chromosomes stop moving. Chromosomes will then uncoil and loosen back into chromatin. The nuclear envelope will start to reform, as you can see here. The nucleoli reappears here. The spindles are completely gone, and the cell is almost completely divided.